stress the cell, you do not have any clear localization of NRF2 in the nucleus. So this explains why the SOD enzyme were not induced, which also explains why the superoxide were so toxic in frataxin-depleted cells. So, we then thought that maybe we can find compounds that will reactivate the NRF2 signaling pathway, or at least interfere with the signaling for SOD enzymes. And among the family of compounds that are able to do this, you've got the ligand of what is called the P-pair receptor. P-pair are molecules which are present at the nucleus membrane, and which, upon the fixation of a ligand on the protein, migrate into the nucleus with their ligand, and that again are able to bind to different genes and increase the transcription of the gene. Interestingly, one of the P-pair, the P-pair gamma, is able to induce antioxidant enzymes and proteins, including the mitochondrial SOD. So we went to the next step, which was to know if there will be a ligand that could do that. And by luck, research is often luck, we found that Pioglitazone is one of these molecules that is able to bind to the P-pair gamma receptor and is able basically to do all what P-pair gamma ligand do. It's, let's say, increase antioxidant enzyme in defense in the cells. The situation is much more complex because when you bind something to a P-pair receptor, you've got much more targets that you can imagine first. You've got many targets that depends on tissue, on conditions, on co-transcriptional factors. So it's very complex. It's very difficult to predict what you will do with pioglitazone by treating cells. You know that it will include a lot of mitochondrial enzymes, a lot of antioxidant enzymes, so basically it has all what we need, but a lot of things can happen. But because pioglitazone was used without too much side effect, we thought that probably it was one of the good ones to be tested. And this was more or less reinforced by different observations that were done after our suggestion. So for example, you've got one or two studies that confirms the data from transcriptome analysis that previously have found that frataxin itself was indeed induced by ligands of the P-pair. Because, with no surprise, because frataxin is one of the mitochondrial enzymes, in addition clearly related with the oxidative defense, so no surprise that it could be stimulated when you bind something to the P-pair receptor. Then there is another observation that might be of interest also in terms of pioglitazone, is the fact that the molecule is able to bind to some iron sulfur cluster directly. Now we speak on another mechanism, no relation, a direct effect of pioglitazone on the iron sulfur cluster of one of these proteins is named mitonite. We don't know too much about the function of this protein. We know that one of the pathology is involved in some human disease, but the idea is that, yeah, maybe pioglitazone, in addition to bind to P-pair gamma, could maybe interact with some iron sulfur cluster of the cell and stabilize this iron sulfur cluster. And then there is also something that came in my hand just at the moment where I was thinking of pioglitazone, which is this paper. I don't know exactly the value of this paper, but it's only a report on one case. But it's the observation is that by treating a patient with multiple sclerosis with pioglitazone, you can have, at least in some cases, spectacular effect. And this told me that pioglitazone was able to reach the brain and the neurological system, and at least to provide some defenses to the neuron, even if neurons have very low amount of P-pair gamma receptors. This study, in fact, on multiple sclerosis was accompanied by a lot of study on animals. And different models of animals again show that pioglitazone might be active under various neurological conditions. And so again, you see here, now it's the muscle strength increased, the body weight, the lifetime, the decreased inflammations, and motor neuron mortality was reduced in ALS mouse model.